Hi, in this video I am going to talk about genetic level inventories. Genetic level inventories are actually a subdivision of species inventories. In this, we are going to study individuals in a population. It is just like the genetic diversity and species diversity. We are actually studying diversity within a species. So genetic level inventories are comprehensive, has the comprehensive genetic information about a species, basically population diversity within a species. So we are studying how diverse a species is. It is more in-depth study than the species diversity. So population diversity studies are especially important for conservation of a particular species. And genetic level inventories are made only for few species because it is expensive and it's very difficult to maintain. Actually, in order to get a genetic level inventory, there should be molecular and quantitative genetic variation should be measured. The molecular diversity measurement is actually done by molecular biology techniques, and the it should be quantitative also. We cannot take few organisms and said this is the genetic diversity, because in order to make a quantitative analysis, the more members of the population should be analyzed for genetic level inventories. So the basic aims of the genetic level inventories are: first is conservation genetics. So for of the, some of these endangered species like maybe panda, tiger or blue whale, these population of these individuals, these endangered species are decreasing dramatically. So if you want to know why it is decreasing so fast, we should have an idea about its genetics. Most times it may be due to the genetic drift or inbreeding. So we need to find the reasons for that. Once we know the reasons for that decline, then only we can make some ways to conserve or manage its population. So one of the main aim for genetic level inventory is for conservation. And it is done only for the species which is under endangered or threatened category. Second reason for doing genetic level inventory, to find significant genetic variation. Sometimes some of the genetic variations has beneficial for us, like some of the genetic variations are uh, beneficial for the environment, or it may have some social values or scientific values or economical importance. Some things like we are finding a new medicine, or we are making a new commodity for the market. So identification of such type of genetic variation is also important. So we are using the genetic level inventories basically for conservation and finding the significant genetic variations beneficial for humans. So in genetic inventories, we are actually documenting the genetic diversity within a species. So how different the members of a species is. If we are going to make a inventory, genetic level inventory for tiger, we are actually measuring how different the populations of the tigers are. What are the properties of tigers seen in Kerala, and what are the properties of tigers seen in West Bengal. So we are genetically studying these tigers by using molecular biology techniques and comparing their genomes to see that how they are similar and how much they are different, which will actually help to make some breeding arrangements which will be helpful for the conservation. And this genetic diversity is identified actually by genetic surveys. It's actually we are capturing the genetic informations from by taking different samples from this organism. If you want to make a genetic survey of tigers, you need to get some samples from the tiger, and there are so many non-invasive methods are there. You can actually make the genetic material by taking the fur of the tiger, so that will be enough to make genetic informations. And these are the methods which can be used to make genetic diversity inventories. First one is DNA sequencing. Actually, sequencing the whole genome is practical nowadays. Uh, so the best method will be DNA sequencing. If you want to sequence the whole genome and see how the whole genome of the two different members of a species is different, that will be actually very useful, and it will be insightful to see many differences and similarities between these genomes. But it is not that practical to sequence whole genome. But most of the time what people do, we will take some selected genes the genes which are actually indicating the diversity, and we will sequence only that particular genes and compare across other members of the species. So DNA sequencing is the best method. It's actually finding out the sequence of nucleic acids in DNA. Second method is to MHSC assay. MHSC is the major histocompatibility complex, which is associated with the immune system. So the similarities, the differences within a species can be easily identified by studying the variations in MHSC. Next is mini satellites and microsatellites. As we know, most of our DNA are non-coding DNA, and this non-coding DNA is consist of repeated DNA sequences. So these repetitive DNA sequences constitute the mini satellites and microsatellites. If you study the difference in the mini satellites and the microsatellites in a species, we can actually get an idea about the diversity. Or we can go for RAPD, random amplified polymorphic DNA, which is extensively used in studying the diversity in plant species. So these all techniques are molecular biology techniques, which can bring out the genetic diversity of a species. The genetic diversity inventories are difficult to make. I already said that. The thing is that making a species level diversity itself is difficult. And most of these genetic level inventories are supposed to be made for species which are endangered. We need to get the samples, we need to do the sequencing work, which is expensive and time consuming, and we need to do so many things to get a complete genetic level inventories. If there is only maybe 100 or 200 members of species living on earth, that it's practical. But in situations like blue whale, it's very difficult to find them, or situations like tiger, it's actually dangerous to find them. So there are so many considerations when we are trying to make the genetic level inventories. So for getting a overall idea about genetic diversity in a particular ecosystem or a forest or something like that, there are some methods which is basically called rapid biodiversity assessment. It actually measures the diversity, overall diversity in a geographical area, without like spending more time there. By looking at some of the indicators, we can actually predict the overall genetic biodiversity. So overall genetic biodiversity of a forest like Amazon or something like that is very difficult to identify, because there are so many different types of species there, and most of the species are unidentified or unclassified. So how do we study the genetic diversity of unidentified species? That's a bit of a question there. So we have several methods, which is actually not a absolute measure of biodiversity, but which actually make the best guess. So those include, first one is higher taxonomic level. In this method, we are studying only organisms at a higher taxonomic level. Instead of studying how much diversity there in a species, we are studying how much diversity in an order, or in a family, or in a genus. And based on that measurement, we are projecting our data to the species level diversity. So, in a forest, you are looking at how much different family members are there. If you are looking at the animals, most of the like many animals will be belongs to a single family. So you will count the number of families and represent the data as the family level biodiversity, or the order level biodiversity, or even the genus level biodiversity. uh, but in this method, relative distinctiveness is not represented. The species level data is not there, or the individual level data is not there in higher taxonomic level biodiversity assessment. But the thing is that it is easy to do, and it is doable. Second method is taxonomic surrogacy. Taxonomic surrogacy. In this method, we are actually studying the biodiversity of recognizable taxonomic unit, or RTU. These species are called morphospecies. The peculiarity is that we can actually identify or measure the diversity of the species by just looking at the morphological diversity. You don't have to jump into the genetic diversity, but it will make a best guess about the genetic diversity. If an organism is morphologically diverse, there should be genetic diversity will be there. But it's a crude type of measurement, but easy to do. And it's actually an indicator of species richness. There is a direct relationship between the diversity of recognizable taxonomic unit and the overall species richness. Most of the time, we will get reliable data on that. But we have some of more reliable methods like taxon focusing. In this method, we will find some umbrella taxa or indicator taxa. These are just like indicator organism. Um, this organism will act as a surrogate for wide range. If you study the diversity of this particular organism, you can actually measure the species richness of a wide variety of species. It is so related. It's like the keystone species in a community or an indicator species in a biodiversity. So in taxon focusing, we are focusing only on a particular taxa, but this taxa, diversity of this taxa is directly proportional to the overall biodiversity. And we are studying we are conducting a very detailed study of this particular taxa. We are not studying any other organism. We are studying only this particular taxa, but we are carrying out a detailed study in limited number of carefully chosen taxonomic group. So that's also doable. We are not studying the diversity in a huge group. We are studying in a small group, but it's a very detailed study, and it is very useful for conservation policy making and management. It's actually give an idea about the total biodiversity. And when we compare this taxon focusing with the previous method, taxon focusing has many advantages. It's more scientific, it is more reliable, and it give more dependable data. One of the example of taxon focusing is use of the tiger beetles, which can actually say the overall diversity of the insects in a geographical area. So the studying insects are always difficult because it is very difficult to find and it is more diverse. So instead of studying all insects, we are going to study the tiger beetles. So by studying the diversity in the tiger beetles, it acts as an umbrella species for another all other insects and give a better idea about the overall species diversity of species. Finally, we have molecular taxonomy. It is like a most advanced and sophisticated taxonomic system. It is based on operational taxonomic unit, so we call it as molecular operational taxonomic unit. And in this, we can actually identify the organism based on the molecular taxonomy, and we can actually identify the biological properties without it going into a detailed studies. In traditional system, if you find a new organisms or if you get a new species, you need to carry out a very detailed analysis to establish that this organism is a new one, not an already existing one. There should be a morphological analysis, physiological analysis should be there, life cycle studies should be there, and it will take like 5-10 years to complete all these studies because the life cycle may be too long. Maybe the whole life, the organism will live for 20-25 years. If you want to study everything, like it's like all stages of life, it will be really difficult. So in molecular taxonomy, what we do, we will directly identify the DNA and sequence the DNA. Once we have the sequence of DNA, we will compare this sequence of DNA with all other known DNA sequences. And based on similarity, we can establish it as a new species or an old species, and we can also establish the evolutionary similarities between these organisms. The molecular classification can be done, naming can be done, so everything can be done very quickly. So that's the advantage of molecular taxonomy. It helps us to taxonomical identification, as well as the biological properties can be also be studied, because based on the similarity studies, how it is similar to already existing species, we can study the biological roles and ecological roles also. uh, and for this molecular taxonomy, we need only single specimen. We don't need to get the whole organism to study its anatomy and physiology, everything. If you want to study an organism, a hair follicle is enough to get enough data of the whole organism, because the hair contains cells and it contains DNA, and right now we have good technique to isolate the whole DNA very accurately, and we can actually sequence it, compare it, and do everything. So this technique can be also used to identify morphologically indistinguishable taxa. If two different taxonomic species are look alike, but if you took the DNA, it will be so different. So we can actually identify the morphologically indistinguishable taxa by using molecular taxonomy, and this single technique can be used for all specimen. In most of the cases, when we are identifying a microorganism or animal, we are using a totally different strategies. But in case of molecular taxonomy, the same technique, the isolation of DNA, the sequencing, comparing, the bioinformatics stuff, everything is common for every organism. So we don't need specialist in all field. We need a specialist in molecular biology. He can actually identify all organisms based on the molecular taxonomy. So these are the techniques used for the genetic diversity studies and the genetic level inventories. And thank you for watching.